Okay, muscle tissue in the muscular system. All right, so we have three types of muscle tissue. We covered this briefly, when we looked at the tissues chapter. We have skeletal, cardiac, and smooth muscles. Skeletal muscle has very long multinucleated cells with lots of mitochondria, right? It has a striated appearance, and it's under voluntary nerve control. Primarily attaching to skeletal bones. There are a few areas in the body where we have skeletal muscle that's under volitional control, but does not necessarily attach to bones. But on the most part, for all intents and purposes at this point, this is the kind of muscle that attaches to bones. So when it shortens, it moves the bones that it's attached to. Right, cardiac muscle has one nucleus with gap junctions and desmosomes. These are located at what's called intercalated discs. This is unique for cardiac muscle. It too has a striated tissue appearance, but is under the autonomic nervous system control, so it's involuntary, and one location in the heart. Smooth muscles are very short cells that have what's called a fusiform shape, kind of like a scar shape, with one nucleus. They are not striated, because they're not long enough to be striated. They're under autonomic involuntary control. And they're found essentially every place else that we find muscle that doesn't necessarily attach to bones or is located in the heart. So places that we're going to see it, especially in AMP2, is going to be the muscle that lines and surrounds the digestive system, the urinary bladder, even the ureters, blood vessels, the uterus. So any place else other than essentially attaching to bones or found in the heart. So muscle functions, it produces movement of the body and fluids. So skeletal muscle, certainly the body, because it moves the bones, at least generally, at least one end of the bone that it's attached to. When it comes to moving fluids in the body, the heart pumps and pushes blood through the blood vessels and through the heart itself. Skeletal muscle helps maintain body posture, stabilize joints, and generate heat from contractions. And that's mostly from a skeletal muscle source. All right, I love these little charts because these help you give something to, or a method of repeating and drawing over and over again, maybe on a whiteboard, so that you can compare and contrast skeletal, cardiac, and smooth muscles by location, appearance, control, number of nuclei, speed of contraction, and then other features. So here under skeleton in green, location on bones, striated appearance, nerve control voluntary, multi-nucleated, and the speed of contraction is very fast. All right. Cardiac muscle only found in the heart. It too has a striated appearance. And now the nerve supply is involuntary through the autonomic nervous system. It generally has like 1 to maybe 2 nuclei. Its speed of contraction is medium. And unique features is that it's autorhythmic and it contains intercalated discs. So at the intercalated discs is where we find the gap junctions and desmosomes. And autorhythmic because if we don't send a signal, down these nerves, let's say the cardiac acceleratory nerve, to the heart, it will pump on its own. And it has 3 different locations where it can generate its own heart rate, starting at the SA node to the AV node, to the Purkinje fibers, so that, the heart will continue to pump, even though you might not send a stimulus to do so. But we look at that again in the cardiac chapter in AMP2. Smooth muscle is found in blood vessels and other organs. Its appearance is smooth. It too is under involuntary nerve control, 1 nuclei, and the speed of contraction is slow. So I really love, and if I create these charts, I generally ask questions from it. So I would, this kind of puts everything in one page for you to study from. Okay, so skeletal muscle gross anatomy. Well, we have cells, often referred to as fibers. but I'll usually refer to it as a cell so that the F word is just going to be used for fascicles. But we have cells that are surrounded by their own connective tissue layer called the endomysium. Endomysium. Endo means within, mysium is muscle. Then the cells in these fibers get organized into bundles called fascicles that have their own connective tissue surrounding them called the perimysium. And then fascicles come together to be organized in one entire muscle. You know, let's say your biceps brachii or your brachioradialis. And the connective tissue that surrounds it is the epimysium. Epi means above and around. The epimysium continues as the tendons that attach muscle to bones. It creates the origin and insertion. All these three connective tissue layers are interconnected to one another to transmit forces to its attachment sites. So, you know, when we look at these, models, especially in lab, and then the pictures here in lecture, try to picture the interrelatedness. Within one cell or fiber are going to be, even within that one cell and fiber, these repeating units of actin-myosin, these myofibrils. And then they get packaged into like one cell or fiber. Then cell and fibers become packaged together, and all their endomysiums of all the cells and fibers kind of coalesce and form one entire perimysium, kind of bundling them into one fascicle. And then all the fascicles, perimysiums, kind of coalesce to form the epimysium, which then continues as the white tissue, connective tissue that we see on models and in diagrams that attach the muscle to two different bones. So that as one, cell and all the different myofibrils within a cell start to contract and shorten, so will all the cells in the fascicles, all the fascicles in the entire muscle, that ultimately, as they're coordinating, shortening and lengthening and shortening and lengthening, that the forces are going to be transmitted to the tendons, to the two different bones. Generally, one bone is immovable, and one is going to be highly movable. All right, so here we look at, this telescoped out, whichever direction you'd like to start. If you want to start the outer layer, the epimysium, we see that in the white connective tissue. It's going to be dense, regular connective tissue. You see how here we have the tendon that's going to attach to the bone. In this case, it's a femur. Then when we look within the entire muscle, we can see these bundles called fascicles that repeat all over. And so surrounding each fascicle is more that white tissue that is all interconnected to one another, from fascicle to epimysium. And then when we look within a fascicle, now we see we can pull out one muscle fiber or one muscle cell, multinucleated, and you can see all these other, it looks like a little box of uncooked spaghetti. Very parallel fibers with it. These are these myofibrils, right? So even within one cell, we have repeating units on end. So we have the endomysium that surrounds that one muscle fiber, and then it continues white to the perimysium and all the whites to the epimysium to the tendon. Right, so the tendon is a cord made of dense, regular connective tissue that attaches muscle to the periosteum of bone. It's extensions of the epimysium and ultimately the perimysium and the endomysium. So what we're saying is that they're just all interconnected to one another. An example of that is an Achilles tendon that can, holds the gastrocnemius to the calcaneus muscle, calcaneus bone. Okay, so nerve and blood supply. Every skeletal muscle fiber has a motor nerve ending, which stimulates the fiber. So this is going to be different than cardiac muscle. We have a certain amount of nerve supply to the cardiac muscle and to your heart, but it has a different way of communicating the impulse to contract from cardiac muscle cell to cardiac muscle cell. But in skeletal muscle fibers, we have a motor neuron ending to each fiber. It has one artery and one or more veins to carry waste away. Arteries supply oxygen and nutrients. Veins carry carbon dioxide, waste products, and metabolism, as that skeletal muscle works. A motor unit, it's groups of skeletal muscle fibers stimulated by one motor nerve. It's the ratio of neuron to muscle cells. A small motor nerve, we find small motor units where we have small muscles that are associated with precise movement and control versus large motor units for gross movement and large muscles. So let me kind of explain what we're referring to here. You have a motor neuron that can branch off at its ends, at its axon terminals, to stimulate maybe 10 or 20 skeletal muscle cells, or it might branch off and stimulate 100 or 150 muscle cells. Where we find this small motor unit of one neuron to, let's say, 10 or 20 muscle cells are going to be muscles like the muscles that attach to your eyes, so that when you're reading and looking in different directions, we have very precise movement. The movement in your fingers to carry out different activities versus stimulating maybe your quadriceps muscles or your hamstring muscles or your pectoralis major, your rectus abdominis muscles. These are large muscles, and they don't have precise movement. They have very large, massive gross movement. So you might have one motor neuron communicating to 100 or more skeletal muscle cells or fibers. Now, this picture is from your textbook that's trying to indicate the difference between the two motor units. I would say the motor unit 2, isn't a very good example. Because it's common for a large motor unit to communicate to 50, 75, 100 different cells. So I would have shown that in the image, but I think you get the point. This picture over to the right-hand side, I love this. If you get a chance to look at some of the slides in lab, this is what we call the neuromuscular junction, where the nervous system meets the muscular system to communicate from one system to another. It's a very important interface. So in the black background, it kind of looks like spider legs here. That is a motor neuron, with its axons and its axon terminals, where we store neurotransmitters. In this case, it's going to be acetylcholine. And then the orangey parallel fibers all to the background, that is going to be your skeletal muscle. Sometimes it's in this kind of orangey, yellowish kind of color. Sometimes in the more pinks, in red kind of color. Okay, so the microscopic anatomy of a muscle cell. So we start with the sarcolemma, because we'll talk about one cell, right? And instead of referring to the plasma membrane of a skeletal muscle fiber as a plasma membrane, it has its own name called a sarcolemma. So it's just a fancy name for a plasma membrane. And of course, it has receptors on the surface, for communication between different cells. So sarcolemma. The sarcoplasm is the cytoplasm containing glycogen and myoglobin. Glycogen, remember, stores glucose. Remember, we said that glycogen is the macromolecule form, the polysaccharide, short-term energy storage of carbohydrates from the chemistry chapter. And myoglobin is your oxygen-storing pigment. So what we're saying here is because skeletal muscle is going to contract and relax and contract and relax, and has multiple nuclei and lots of mitochondria, it's going to need that because it's going to contract. We need a lot of ATP. And we said that the mitochondria, produces ATP through cellular respiration. Well, to do that, we need a substrate called glucose, or maybe even fats, and we need oxygen available. So we actually can store excess glycogen and myoglobin, oxygen, in skeletal muscle cells, so that we have immediate storage, and resources available for contraction. We have transverse tubules. So the transverse tubules are actually a continuation of the sarcolemma, right? And they travel to the interior of the cell, carrying impulses from the surface of the cell deep into the cell, so that we can get, coordinated muscle contraction. The sarcoplasmic reticulum is a specialized endoplasmic reticulum for calcium storage and release. When we looked at the composites, cell models, and we looked at the RER and the smooth endoplasmic reticulum. This is a type of a smooth that's going to store and release calcium. Then we have our myofibrils, which are the bundles of overlapping contractile filaments that extend the length of the muscle. It contains actin and myosin. Actin is going to be the thinner fibers. Myosin is going to be the chubbier fibers or proteins. And that's what gives skeletal muscle striated appearance, as well as cardiac muscle, because it too is striated. All right, so I think it's easier to start at the bottom, where we look at one of those muscle fibers or cells that's kind of telescoped out. So we go, okay, to the outside is the sarcolemma, and superficial to that and surrounding that would be the endomysium. All right. So if we took the endomysium off the connective tissue layer, we would have the plasma membrane or sarcolemma of the muscle cell. And when we looked at it in that previous diagram, I said, it looks like this. It's kind of a bundle of uncooked spaghetti, all stacked up. Well, those are the myofibrils containing myofilaments. Right? So not that you really have to distinguish between a myofibril and a myofilament, but we're just going to show you what it looks like. All right, so if I pull out one of those pieces of what I call uncooked spaghetti, we're going to see these blue areas, these red areas, these white areas in there. All right, that is the repeating units called sarcomeres along one of these myofibrils. So that's in between from blue to blue to blue stripe on that myofibril. If we magnify it and we look up at the top, then we're seeing kind of what that looks like. You can see in white in the vertical lines, kind of in pairs of lines there. Those are your striations. You can see all the nuclei there. Right here we have one fiber, right, that we telescoped out, or one cell. And you can see those repeating striated fibers. Okay, so now over to the left-hand side, upper left-hand corner, we're looking at sort of that same picture, all right, from the bottom here. All right, we're going to pull out a section. So we're going to look and see, well, what is actually, what is composed there? We have the sarcolemma to the outside, which is indicated in white, and because the transverse tubules are a continuation of sarcolemma, they too will be white. Okay, so over to the right-hand side, we're going to look and see what those, what it might look like, all those myofibrils stacked on one another. So to the outside in white, we have the sarcolemma. The white tubes coursing to the interior are the transverse tubules. And down on the bottom, it shows you that they're sort of like a tube. And the reason why is because when we generate this impulse, this electrical kind of signal on the sarcolemma, it's going to travel down the middle of that transverse tubule, be carried down kind of in that hose, so to speak, to the interior of the cell. Now that transverse tubule sits adjacent to and on top of these blue sacs here called the sarcoplasmic reticulum. Where you have the two sacs and the transverse tubule come together, those three structures, it's called a triad. It's those blue sacs that are going to store and release the calcium. Right, then up on top, they took the transverse tubules and the sarcoplasmic reticulum off, so you can see the blue lines there and then the red lines there. Right, and where you see the skinny, lines in blue, that's going to be your actin, and where you have the chubbier lines, that's going to be your myosin. All right, so the microanatomy of those myofibrils, the repeating unit is a sarcomere. Right, it's a functional unit, the area of a myofibril between these what's called two Z discs, and they overlap, or the overlap of filament bands produce a striated appearance. So let me just go down one so you get what we're looking at here. All right, so one unit, all right, is called a sarcomere, and you see up on the top it has sarcomere with two arrows, which includes Z disc to Z disc. Right, where you have that zigzaggy line and a zigzaggy line. Within that is one sarcomere. Within that, we have the two blue, looks like two strings of blue pearls, so to speak, or beads. Right, those are your actin filaments, and they repeat all the way down. They don't contact one another, but they extend from the Z disc toward the midline. That's why they call them lemon. And then sandwiched in between those are these chubbier red filaments, proteins called the myosin. And they have what's called myosin heads on them. They kind of look like golf club heads that are sitting on them. All right, so when we're talking about a sarcomere, that's repeating functional unit of muscle, of skeletal muscle. The blue, in this case, the two coiled of the ropes there are the actin filaments, and the red are the myosin filaments. Okay, so muscle proteins, myofilaments are the contractile proteins. This is where we generate force within, or during muscle contraction. Because these two filaments, the actin and myosin, are going to make contact with one another. We're going to show you some videos of that. So myosin contains projections, myosin heads, which will fit into the actin molecule. We'll see what that looks like. There's a little depression in there. They make up thick filaments. The actin proteins contain myosin binding sites where the myosin head is going to attach. These are the thin filaments. On the actin are these two what's called regulatory proteins, right? They switch contraction on and off, or they allow or disallow contraction to occur. Right, so we have these two. It's called the troponin tropomyosin complex. They block binding site on the actin molecule when we want muscle relaxation. And when we want the muscle to be able to contract, they're going to unblock this binding site so that the myosin heads, or the golf club heads that I referred to, can make contact with actin. And then they're going to row toward the center and create and generate force. Okay, so now we're back to some of these pictures again. All right, so here we're saying the two blue, the blue coils of beads, so to speak, the actin, and that repeats 1, 2, 3, 4 times. And then in between, in red, is the myosin, right? The chubbier, thick filaments. So what is going to happen when you want muscle to contract is that the red golf club heads, of the myosin is going to make physical contact with the blue beads there. And then they're going to row toward the center of that sarcomere, bringing the two ends of the sarcomere close to one another. All right, so the sarcomere is going to have a certain length before contraction when it's relaxed, and it's going to become much smaller. That is contraction. And when that happens thousands of times down the muscle fiber, of all the muscle fibers in a muscle cell, all the cells in the fascicle, all the fascicles of that particular muscle that now translate that force to the tendons, you will get movement. All right, so the thick filament is showing you in red down here. Here you have the myosin heads. I think they look like golf clubs. All right, and on the bottom, it shows you how it's kind of a twisted coil as well. And then we have these myosin heads where it's going to have an ATP binding site on it, but don't stress about that. Over to the right are the thin filaments, and this actually lets you see this troponin tropomyosin complex. They're two different molecules, but we just kind of throw them together. So here we have the two coils of blue beads, so to speak. And if you notice, there's an opening on one side. All right, there's a depression there. That's your binding site for the myosin head that can fit into it. Around myosin head is going to be able to attach to that binding site. So everything reminds me of food, I guess, but this reminds me of olives without a pimento in it. That it's a cavity, so to speak. All right, so on top of and overlapping the openings on these two strings of these active molecules is this troponin and tropomyosin complex. The tropomyosin is actually the rope part, and the troponin is the yellow beads. And that is where on troponin the calcium will bind to, so that this molecule rolls and uncovers the binding sites so that myosin can attach there. All right, so currently you see this troponin tropomyosin molecule blocking the openings of all those little actin molecules. But when we want to contract, that troponin tropomyosin is going to move and roll over, so to speak, and uncover the binding sites on actin so that those myosin heads can attach to it. So there will be a few videos we're going to watch that demonstrate this. And the reason why I like these videos is that you can watch it kind of over and over again. Now, sometimes they go into a little more detail than you really need, all right, for understanding this right now. And I appreciate the fact that they're going to be talking about the nervous system, which we cover in the next chapter. But we have to understand a little bit about the nervous system to know what's happening in the muscular system. And we'll get into more detail about the nervous system in the next exam. But we're going to talk about, a step-by-step process that occurs, when we need to have muscle contraction and what's happening, and then how do we stop muscle contraction, so to speak. So if you're downloading, which I would recommend, is downloading these notes to a flash drive, because the videos within these notes, these PowerPoint notes, take a while to download. And if you go into the course page and you go to download it, so you can watch it over and over again, it's going to take some time. I just think it's easier to save really all the notes and store them, on a flash drive in the event that, your internet goes out or something, you at least can have access to the notes. But the videos are really helpful to watch over and over and over again. It's going to be helpful to understand that when we get to the nervous system and then further in AMP2. Okay, so that takes us to our first video. Now, I'm going to break the share and reshare it because I believe once I start this, it's probably you won't be able to see it unless I reshare it. So I'm going to stop the share. I'm going to start this and start the share. Let me stop it first. Hold on. Let me stop the share and then try to reshare once the video gets going. See if I can do it this way. I might have to do it more than once. Hold on. See, and I have multiples, so I just have to find the right one. Hold on, let's try screen number 1. No, let's try this one.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, An action potential propagates along the sarcolemma. Neurotransmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in two ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
Okay, so that's the first video. I just want to make sure that I'm sharing the right thing with you. Yep, hopefully you still have the, actually, I'm going to stop the share again and reshare to make sure that you have the PowerPoint. Zoom has some, not the Zoom, but yeah, Zoom has these funny little things that sometimes it connects and doesn't connect. Okay, we should be good. All right, so that talks about how we have the impulse traveling down the motor neuron, right? What are the events? And I know, you don't know what an action potential is, but that's the electrical signal. That's a signal. Travels down to the ends of this motor neuron called the axon terminal. And within that, you have these synaptic vesicles. And remember, vesicles are just kind of like balloons that store something in it. It's membrane bound that contains and stores something. And in this case, it's going to be acetylcholine, a neurotransmitter. All right. So when the impulse travels down that nerve, it stimulates calcium, these calcium voltage gates, to open, and calcium enters that axon terminal. And that's how the cell knows, okay, I need to release the acetylcholine. So those vesicles merge with the membrane and release it into this little gap area, between the nerve and the muscle. They don't actually touch, but they come very close to one another. Acetylcholine travels across the gap and binds to a specific receptor for it. And when that happens, we create another signal on the sarcolemma, which travels down the transverse tubules to the interior of the cell. All right. Now we're going to talk about now, and it's going to be demonstrated what happens after that. So it takes it in groups. You don't have to understand this, or you don't have to memorize this of all the events in one video to a video to a video, but it lets you see them in pieces so that you don't get overwhelmed in like a 10-minute video on this. All right. So now we go on to the sliding filament theory, because we need to go from this impulse is going to travel down these transverse tubules. All right, those transverse tubules sit on top of that sarcoplasmic reticulum storing the calcium. Well, that impulse traveling down the transverse tubule will stimulate the release of the calcium from those blue interconnected sacs. The calcium is going to travel to the troponin and tropomyosin and bind to it, so that it rolls off those binding sites on actin so the myosin heads can make contact with it. And when the myosin heads make contact, I said that they row toward the center so that the ends of the sarcomere become closer to one another, and that's muscle contraction or shortening. That is creating muscle tension. So we create muscle tension by myosin attaching to actin, and approximating the ends to one another. So there's different terms for that, and that's what we're going to cover now. All right. So the movement, when myosin makes contact with actin, the actin molecules are going to approximate one another toward the center. Those blue chains of beads, are going to approximate one another in the center and actually kind of go past one another a little bit. All right, so that's what we call the sliding filament theory. It's the current theory on skeletal muscle contraction mechanism. In a relaxed fiber, the thick myosin and thin actin filaments overlap slightly, but during contraction, actin slides past myosin, shortening the fiber. That's what they call the sliding filament theory, because they're going to slide on one another. After contraction, actin and myosin return to the original position. All right. So the impulse transmission from the nerve to the muscle occurs at the neuromuscular junction, like we just saw. All right. When we have the cross bridge formation, it's going to go through a series of events here. Now they're going to talk about step 1, 2, 3, 4, and I think it's really good to see this. Don't worry about all the steps of hydrolyzing ATP and, when we have inorganic phosphate and then it detaches and so forth. I just want you to see that when calcium binds to that troponin tropomyosin complex, all right, that it moves and the openings on actin become exposed. And now myosin makes contact with it. It's going to row toward the center, and then it's going to take a molecule of ATP to bind to that myosin for it to let go and re-energize itself. It's kind of like putting your oars in the water and rowing, and then taking your oar out and then repositioning the oars behind you to go back into the water again. You're getting ready for another stroke. All right. So this is this cycling of myosin over and over and over again, attaching and unattaching to actin, is called the cross bridge cycle. It takes one ATP molecule for each cycle. Don't worry about the rest of it. I just want you to know one ATP for each myosin stroke like this. So, it takes a lot of ATP for muscle contraction. Okay, so that takes us to muscle contraction part 2. Excitation, contraction, coupling is just a fancy word for all the events that happen after this. All right, and again, just know we'll go over the basics of what you have to know. So I'm going to start this, and then I'm going to stop my share and reshare it just to make sure we are on the video.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with the calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus 2 adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
Okay, so I like that because it shows how the impulse is traveling in the transverse tubules and how we're going to release calcium from the sarcoplasmic reticulum. Love that. Okay, so now we're going into the cross bridge cycle. Cross bridge formation is when myosin attaches to actin, like I just showed you in that other diagram. Right? The power stroke is when you row your oars in the water, so to speak. Right? So when myosin approximates, they row toward the center and they approximate the ends of the active molecules toward one another. That is our power stroke. It generates force as they rotate toward the center of the sarcomere. These actin and myosin filaments, they slide over each other. Detachment of myosin heads requires an ATP to bind to it. All right, so this is when you generate force. Prior to myosin making contact with actin, you got nothing. All right, but once it contacts it and it starts to stroke, like row an oar, then that is the force. Right? Once it lets go, you stop generating force. You have to make contact again and again, have the myosin heads row toward the center. Okay, so that takes us to the next video. Again, I'm going to start this and kind of stop it.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released, and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin binding site on actin.
Okay, so let me just reshare here. So let me discuss something here, is that, evidently the videos are made by a different company than Pearson. Pearson is a publisher, and they create the notes and a lot of the images in the textbook, but they don't always make the videos, I guess. And so sometimes there's like little boo-boos in there. And I wouldn't say it's necessarily a boo-boo, but I will just say this, right, is that, they talked about, which I thought was really good, showing you how the myosin head is going to row over and over and over again. And it says, as long as calcium is binding to the troponin tropomyosin, you're going to get contraction. And, that's true, as long as you have enough glucose and ATP, oxygen and things, as long as you're able to make enough ATP to keep binding to those myosin heads so they can detach and kind of reload again. And the degree of calcium is really controlled at that neuromuscular junction, because when we want the muscle to relax, we're going to stop releasing that acetylcholine, and once that happens, then we no longer send the signal down the transverse tubule. We stop releasing calcium, and now that is the signal for the muscle to relax. And we pump all that calcium back into the sarcoplasmic reticulum and save it for the next contraction. And then the troponin and tropomyosin rolls back and covers that actin so that myosin can no longer make an attachment to it. So technically, to stop further contraction, we stop the release and get rid of the acetylcholine between the motor neuron and the sarcolemma at the neuromuscular junction. That's really how it gets, that's how we stimulate muscle relaxation, essentially, because the rest of that will then get shunted right back into the calcium, back into the sarcoplasmic reticulum. So let's look at kind of step by step, so to speak. We have a nerve impulse travels down the motor neuron to the axon terminal. Acetylcholine is released and stimulates the sarcolemma receptors. The motor impulse travels down the transverse tubules. Calcium is released from the sarcoplasmic reticulum. Calcium binds to the troponin tropomyosin complex, which then unblocks the binding site on actin. Myosin head binds to actin, forming the cross bridge. Then you have the power stroke, right, the rotation of the myosin head, where causing actin to slide over the myosin. That's our contraction. ATP binds to the myosin head so that it detaches from actin. And if we have multiple stimuli coming down that motor neuron to keep releasing the calcium from the sarcoplasmic reticulum, we'll get continued contraction. But if we only want one little contraction, then we're going to stop the acetylcholine from being released at the neuromuscular junction. We're going to break it down with the enzyme acetylcholinesterase. By the way, enzymes end in ASE like that. And the stimulus ends, so contraction stops and the muscle relaxes. And that's really what causes the muscle relaxation. We have to block that neurotransmitter at the neuromuscular junction. So we have a contraction, right? ATP binds to myosin, so it lets go, detaches, right? We pump the calcium back into sarcoplasmic reticulum, so the troponin and tropomyosin complex, recovers that binding site. And we break down, the acetylcholine at the neuromuscular junction with acetylcholinesterase. Now, we can do a couple different ways. We're going to talk about what happens with acetylcholine. So if you're no longer sending these signals down that motor neuron, then, we can have kind of reuptake. We can endocytotically take some of the byproducts of breaking down the acetylcholine and some of the remaining acetylcholine intact back into synaptic vesicles and store them in there, for another signal, as another signal travels down that motor neuron. But if there's just a little bit of acetylcholine there, it will just diffuse away. It's not a significant enough quantity to bind to receptors to cause another contraction. And by the way, if somehow these videos are you're not able to see them, when you download the notes like this and you put them in slideshow mode, they will run for you. So that's the beauty of this. You just have to put it in this slideshow mode like I'm doing, presenting this PowerPoint. All right. All right, muscle tone. Muscle tone is the random mild contractions that keep muscle always in a slightly contracted state. It also stabilizes joints and maintains posture. So what we're saying is that even with skeletal muscle, when let's say you're sleeping, you're lying down, all the muscles or let's say all the fibers and cells of, let's say, your abdominal muscles or your rectus spinae muscles or your leg muscles, they relax, but they don't all relax entirely. I mean, 100%. There's always a certain amount of contraction there. And it kind of rotates different areas. It kind of, one area will contract and then it'll relax, and another area will have a mild contraction and relax. And that's what we mean by muscle tone. A certain amount, a slight amount of contraction, occurring in these muscles. Tetany is called sustained muscle contraction. So if you've ever had a Charlie horse, had a cramp in your gastrocnemius, your calf muscle, you know what I'm talking about. That's tetany, sustained muscle contraction. Muscle fatigue is the inability of muscle to maintain contractions with prolonged activity. So, what's the difference? Well, in tetany, you don't want the muscle to keep contracting, but it's contracting, right? Like a muscle spasm like that, where it can be pretty painful, right? But in muscle fatigue, let's say, for people who go to the gym, or if you're a runner, if you're a cyclist, and you're pedaling or you're running as fast as you can because you see the end, the finish line, and you're sending signals to pedal, 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 or run, run, run. And you're going as fast as you can, but you're burning through all your glycogen stores, your energy, your oxygen, right? And you're breathing heavy and your legs are just slowing down, more and more and more. Or in the case of, let's say, you're doing maybe a bench press, if you want to think of it that way, and you're working your chest, your chest muscles, your pectoralis major and minor. And you get to the point of like, you want to push one more rep out and you're just like, oh my God, I'm barely getting that, and your arms are shaking. That's fatigue, the inability to have muscle contraction over prolonged activity. Why is that? Because you've depleted calcium, creatine phosphate, oxygen, glycogen. You have a buildup of lactic acid, all contribute to fatigue. So that burning sensation in your muscles, it means that you are producing lactic acid because you don't have enough oxygen being delivered to the muscles so that you can convert glucose or proteins or fats into ATP. And when you don't have one of these oxygen acceptors, these oxygen certain stages of this process, you produce lactic acid. Oxygen debt is the additional oxygen taken into the body after exercise used to now convert lactic acid back to some glycogen in the liver and replace creatine and phosphate with ATP in muscles. All right, so what does that mean? So if you keep trying to run as hard as you can, let's say you're in a 5K race or a 10K or whatever, if you keep trying to run at the pace that you want, you are going to fatigue. You start to slow down. Maybe you've timed it such where you're just maxing out at the finish line, but let's say in the event that it's still a ways away and you're running as hard as you can, full bore, is that you can only maintain that for so long because you deplete your calcium, your oxygen, your glycogen, your energy sources. And by the time you get to the end, you're breathing heavy, right? You keep breathing heavy like that until you're able to replace a lot of what's called this oxygen debt. And you can often keep walking some and kind of shake it off, so to speak, or pedal if you're on a bike, and spin some, and while you are taking on this oxygen. And as you start to replenish the oxygen, your respiratory rate starts to go down, right? You're not breathing quite so heavy. And if you keep your legs moving some, your legs don't feel so heavy, do they, right? Because they're so full of blood. But that also allows you to now convert that lactic acid back to an energy source called glycogen to be stored in the liver. That's the oxygen debt. All right, so homeostatic imbalances, rigor mortis, I love this. 3 to 4 hours after death, muscles begin to stiffen. Peak rigidity occurs about 12 hours post-mortem, so after the person has died or someone has died. Intracellular calcium levels increase because ATP is no longer being synthesized. So calcium cannot be pumped back into the sarcoplasmic reticulum. So the calcium is sitting out there, right, able to bind to the myosin, right? This results in the cross bridge formation, all right? So even though you don't need muscle contraction, we don't have the ATP to pump the calcium back. So it's binding to the myosin, or to really, the calcium is binding to the troponin tropomyosin. It uncovers the binding site on actin, so myosin can make contact with it. All right. However, it rose toward the center, right? It has the power stroke, but then it can't let go because we don't have the ATP available and being made. Right. So ATP is also needed for cross-bridge detachment. Results in myosin head staying bound to the actin, causing the constant state of contraction or rigidity. Muscles stay contracted until muscle proteins break down, causing the myosin to release. So what eventually happens is those proteins just start to break apart. You have a very acidic environment. We have denaturation occurring of those proteins from a quaternary structure to a tertiary to a secondary. And because these are proteins, they're just going to break apart. And then you will have more flaccid muscles again. Okay, and that is the end of the muscular system.